Hello, my name is Antti Vuolanto and I present Herantis Pharma here today. So let's first go into Herantis. Most of you probably know, but let's have a short recap. So Herantis is a public company established already 15 years ago, being public for 10 years. And we develop disease-modifying therapy for Parkinson's disease and other neurodegenerative diseases. Our lead asset that we call as HER96 is a small peptidomimetic molecule that combines the unique mechanism of action coming from a natural protein called CDNF, and we combine that with the ability to use subcutaneous injection to deliver the drug into the brain to the target tissue. This year, we were able to complete the first clinical study with HER96. It was a phase 1A clinical study, and there we met the primary and secondary endpoints, and the highlight is that we were able to demonstrate the blood-brain barrier penetration in men. Today or now I will present you a bit about the science and the preclinical evidence that we have. So first of all, what HER96 is. So it is a fragment of the natural CDNF protein. So it mimics the active side of the protein. It is not a natural peptide. It is chemically modified to be a better drug than a natural sequence. So it's a synthetic molecule, which is of course very beneficial, for example, for the point of manufacturing. What we aim to do with HER96 and also earlier with the CDNF protein is to address a mechanism, a pathway called unfolded protein response pathway. And that is responsible of regulating cell stress. So what we want to affect is the cell stress that is causing the symptoms of Parkinson's disease. And we can combine that now with the patient-friendly administration with subcutaneous administration. With HER96, we have earlier presented very nice and very compelling evidence of preclinical activity, the efficacy in preclinical models. We have done the required preclinical safety studies, and now, as I said, we have been able to translate the blood-brain barrier penetration into men in this phase 1A study. So what we aim with HER96 is to improve the current treatment by providing the possibility to stop the slowdown or even stop the progression of the disease. So currently, Parkinson's disease can be treated to treat the symptoms, but not to affect how the disease is progressing. And the disease is caused by the degeneration of neurons in a certain area of the brain, resulting in a decreased amount of dopamine in that brain region, which results in the compromised motor coordination, as those neurons are responsible of the motor coordination of men. The market opportunity is huge. So currently the Parkinson's disease drug market is around 5 billion. So the symptomatic treatment, but with being able to deliver a disease-modifying drug into the market, it's estimated that the market size will at least double by the end of this or by 2030. So what kind of evidence we have preclinically for the CDNF? So one of the key factors for Herantis' success is that we have 15 years of experience with the same biology. And we started with CDNF, we took that through preclinical studies, we went into clinical study with CDNF, and now we have transformed into HER96. And with CDNF preclinical studies and the clinical studies, it's good to remember that we had to administer the drug intracranially, so directly into the brain. And now in this slide, on the upper part, you see data from rhesus monkeys, which had a induced disease like Parkinson's disease using a toxin. And in this figure that you see on the right-hand side on the top, there are like sections of those monkey brains. And there you can see that using CDNF treatment, we have been able to increase the number of the neurons and their axons, so their activity, dopaminergic activity. So actually the neurons are not, we are not able to in a way create new neurons, but we are able to regenerate those neurons that have lost their ability to function. And that has turned into 50% decrease on the motor symptoms. So very clear evidence on the animal side. And the lower part is from the clinical study that we did with the CDNF. The graph that you see, a bit small one, but the essence is that individual that we show here was treated with placebo for the first 6 months, and there wasn't a change in his behavioral motor score. But when we transformed in the next 6 months into CDNF treatment, we saw a big drop in the UPDRS motor scores. Actually, it dropped by 50% during those 6 months of treatment. And then we observed after the treatment that it had a long-lasting effect. And the a tiny gray line represents the standard of care treatment, levodopa. As you can see, the dose of levodopa had to be decreased when we treated the patient with HER96, demonstrating that we were able to affect on the disease. So we want to address the unfolded protein response pathway. And there we have like 3 different aspects that we want to affect. First of all, the neuronal cell stress, then the accumulation of alpha-synuclein aggregates, and then neuroinflammation. And they form a vicious cycle and they strengthen each other. And we want to break this cycle. And now if we can have the video here. So treating animals with the subcutaneous administration of HER96. The first mouse was just a normal aged mouse. The second one has the pathology and is really struggling with walking over the bar. And the third one has been treated with HER96. And as you can see, the third guy is actually walking quite smoothly over the bar. So we were clearly able to affect the motor symptoms of these mice. And this is exactly what we aim with HER96. Just a quick recap of the preclinical data that we have with HER96. So now in these graphs, the white bar represents the normal aged mouse. The red bars represent the disease animals, and the blue bars represent the treated animals. And in the first graph, that shows that we are able to rescue at least 60% of the dopamine neurons in the affected area by using subcutaneous administration for 4 weeks with these diseased animals. And not surprisingly, this results into normalized level of dopamine in the brain area. So we are able to affect the disease. We are affecting the disease in such a way that we have normalized amount of dopamine, which results in normalized motor function. This is just another representation as about the video, similar to the video as you saw. But in addition to that, we are able to decrease the amount of alpha-synuclein aggregates by 50%, which turns into complete removal of neuroinflammation. And this is why we are so enthusiastic about the potential of HER96. So this year we have completed the first in human clinical study with HER96. It was a single ascending dose study in healthy individuals. We had 6 dosing cohorts with healthy young males from 10 to 300 milligrams. And then we had a secondary part with elderly individuals for CSF sampling, so measuring the blood-brain barrier penetration potential. On a high level, the safety aspects, we didn't see anything special there. Some local side reactions due to the injection, like if you get a vaccination, and this allows us to move forward with the formulation and with the administration route. And then I skip these slides and go directly into the essence, which is the blood-brain barrier penetration data. And from the 12 elderly individuals, we collected a single CSF sample to measure the concentration of HER96 in that. And what we can see is that when the sample was taken between 4 and 12 hours, the concentration was between 50 and 100 nanograms per ml, which is clearly on the pharmacologically active range based on the data that we have from preclinical settings. At 2 hours, a bit lower, but that's the reason is that it's still like increasing. So the HER96 first goes into bloodstream, from where it goes to CSF. Of course, we were extremely happy about the result. This demonstrates that yes, using subcutaneous administration, we are able to deliver HER96 into the central nervous system. So basically, the study endpoints were met. And what we do next, so next in our clinical development, we are setting up a phase 1b study to study multiple doses in Parkinson's patients. At the same time, we are preparing other aspects to be ready for phase 2 in 2 years' time. Of course, we also would like to explore HER96 in other indications, as the mechanism of action is not specific to Parkinson's, but it applies also to many other neurodegenerative diseases. And what we really would like to get on board is to find a global development partner. These discussions are now like boosted by the great results, and of course, until a deal is done, it's not done. So we are working towards getting one, hopefully rather sooner than later. Hopefully much before the starting of the phase 2 study. All right, that's everything I had. So maybe we'll continue with Antti.
Thank you, Antti, and please have a seat. Okay, first question from the chat is sort of an entertaining one. So what needs to happen for Herantis to be the next superstar stock in Helsinki Stock Exchange?
A good partnering deal. I think that would do it pretty well.
Yes.
Giving the external validation that yes, the science and the data that we have is as great as we believe it is.
Good. Okay, so let's go to the partnering part. You also mentioned a little bit about this at the end of your presentation. So you have mentioned previously undertaking partnering discussions. So can you talk a little bit more about what has happened recently and what these recent results have kind of changed that landscape?
Yeah. So basically, the first time we discussed about the new data was a month ago at BioEurope, which is the biggest partnering conference in Europe. And there we were like we met like 20 different companies, and the let's say the impression of those discussions with the big pharma was completely changed from the previous years. So the companies were really enthusiastic about what we have been able to achieve in 2 years' time since nominating HER96 as the lead compound, and we were really much endorsed about what we have accomplished, and then of course many of the companies also say that actually currently there are not too many good initiatives, good drug candidates for Parkinson's disease, especially for disease modification, currently available. So I think we are in an extremely positive mode currently. And of course now how we continue from here is that early January we have the JP Morgan Healthcare Conference week at San Francisco, which is maybe the most important event globally. We are invited there by several pharma companies to discuss with their R&D teams. So, of course, this is in a way progressing as we plan. Of course, no guarantee of anything, but at least we are doing things according to what we have planned to be done.
So discussions are ongoing?
Yes, discussions are ongoing.
Okay, and you recently did a share issue of 4.5 million. So what is the scope of this funding? What can you achieve with it? And how does this link to the partnering discussions?
Yeah, so basically before doing the financing round, we had a runway until Q2 next year, which is a bit shortish. And of course now when we go to discuss with the pharma partners, their R&D teams, it's very important that we can show that, yes, we have good support from our investors and that we are not, in a way, back against the wall. We don't need to do a deal and we can negotiate about the terms if and when we are at that stage. So it gives us the freedom to really exploit the data and see whether we are able to have a good deal with this. And of course, we continue the preparations for the phase 2 and the phase 1b, and we can, in a way, continue with the full speed there. But of course, as we have communicated before, 4.5 million now, it's not enough to run the phase 1b or to be ready for phase 2. So at some point of time, we definitely need to have more financing. But maybe the route is via partnering. Let's see.
Yeah. You partly answered this already, but I will still ask, how comfortable are you with your liquidity at the moment?
I think currently we are performing very good there. So the market has been extremely difficult, and raising now additional financing, I think that was really important and actually quite good achievement. So I think now we have like enough time to see how the future pans out, how we can exploit the current data, and I'm very comfortable with the situation currently.
Yes. So there are some other Parkinson's drug candidates in the pipeline. So how does your drug compare to these other candidates?
Yeah, so quite typically the other candidates, they are addressing a certain maybe more narrow aspect of Parkinson's disease. For example, the antibodies that address to remove the harmful alpha-synuclein aggregates, which can be effective and which can be very relevant, but it's only like a part of the story. And we believe that our, let's say, very broad mechanism of action, we can affect many aspects of the disease pathology. We are in a quite unique position there.
And there's a nice bridge to other indications. So how do you see this broad mechanism? Is it applicable to other neurological diseases?
Yes, if we look at the mother protein CDNF, based on which we developed HER96, there is good preclinical evidence on several neurodegenerative diseases with CDNF, so ALS, Huntington's, Alzheimer's, to some degree. So we certainly believe that studying other indications with HER96 would provide, let's say, better value for HER96 and more possibilities for the partnering, and of course optimizing the value of HER96 also for the investors.
Okay, that's all of the questions from our chat. So thank you very much for the presentation and discussion.
Thank you.
And next up we have Aiforia Technologies. So welcome.